Okay, so I'm going to challenge a bit the title of this, one of the words in the title of this meeting, this word inspiration, and argue that what we should be trying to go for more than just inspiration. And that inspiration is a starting point. It's a great starting point, but it's not going to be enough ultimately to get to the point where we want to be of understanding the principles of computation that are actually going on in nervous systems. And that to do this is really a joint endeavor of both engineering and biology. And I say this because I sort of think of myself as living between these two worlds. And in one camp, we're trying to come up with algorithms that can do something useful, that will be of interest to the engineering community and solve problems there. But at the same time, of use to neuroscientists that are trying to probe the nervous system and are searching for good hypotheses of what to look for. And so we want to come up with models that sort of attack both these fronts. And I would argue this is really, I think, what all of us in this room should be trying to do. And I oftentimes hear this kind of, this goal of like, well, I just sort of want to get inspired by the neuroscience, but we don't necessarily need to be biologically accurate. And if we can just sort of get it to do the problem, that's good enough. And I think ultimately it's going to be really involve rolling up our sleeves and getting more into the biology to understand the actual principles at work, as well as coming from the engineering side and trying to drive it more from theoretical principles. And as an example of this, I think I would point to understanding how eyes are designed. This is just a nice illustration from this article by Michael Land and Russ Fernald on the evolution of eyes. And Michael Land has written extensively on animal eyes. And one of the things I think you can see if you look at all these eye designs that have come about through the evolution, the diversity of different eye designs, starting with the pit eye here on the left, going to other eyes like some eyes have pinhole cameras. Other eyes, such as the bottom, use spherical mirrors to do the refraction of light, not lenses, but actually a mirror that does the refraction. And other eyes have lenses that refract the light, and scanning retinal systems, multiple lens systems, and then our eye down here at the lower right. Okay, so I think that one thing that's very evident when you look at all these different eye designs is that you can understand them all in terms of principles of optics. And if you didn't know about optics, you would just be looking at a lot of complexity and you wouldn't understand what you're looking at. And if you were trying to design an optical system, you could say, well, I'm going to try to be inspired by what I see here and sort of make something that kind of looks like that. And you might eventually sort of trip upon the answer of what's actually going on. But for example, the curvature of certain parts of the system is very important, such as the lens, and the curvature of other parts of the system maybe is not so important. And you wouldn't really kind of know what aspects are sort of driving how the system works here. And so now that we know about optics, we can look at all these eye designs and make sense of what's going on in nature. And similarly, I would argue that we're at a similar point now in neuroscience. We're looking at the brain. We see all these marvelous structures under a microscope, and we have no idea what we're looking at. And it's something like a lens, but because nobody's really written down these theories or principles that we're searching for yet. So we're in this interesting point in neuroscience right now, where at the same time we're trying to understand these principles, conceive these new principles of information processing in the nervous system, at the same time as understanding how neuroscience implements them. Okay, so it's really both these things. And so what I'm going to try to focus on here today is just three examples, and there are many more, but three I think, important lessons that we can gain from biology, which I think are they're points of inspiration, but points at which I think we really need to drill deeper and understand on a deeper theoretical level what's going on, as well as in terms of the biological details. Okay, so that is one is how perception and cognition is possible in tiny brains. And then the other is a nonlinear processing in dendritic trees. And finally, feedback on neural systems. So we'll just start with this idea of, so what can you do with a tiny brain? So a lot of our attention, I think, in the community is focused on this beast here, the human brain. And we want to understand all these amazing things that the brain does. It has 86 million neurons and a billion neurons and works with 20 watts. It plays chess. It can formulate theories of relativity. It can also, navigate through three-dimensional environments and so forth. So it does a lot of stuff, and it's not necessarily a good starting point because this is, one of the most complex systems devised by evolution. So we might want to, if we want to understand some of the basic computations, think about starting with simpler systems. And so for example, this dot here would represent some of the nervous systems that I'm going to talk about, comprising less than 1 million neurons and maybe consuming less than 1 milliwatt of power. Okay, so some examples of that are shown here: the jumping spider, the sand wasp, and the box jellyfish. And I'll talk in some detail here about the jumping spider, but the sand wasp, for example, it has amazing navigational capabilities. It builds its nest in the sand, as a little burrow in the sand, and it navigates away from this nest as far as a mile away to hunt prey, basically other honeybees. And it brings those bees back to its nest in the sand, so it can find its way back using various landmarks in the environment, not only from this long distance of a mile away, but it can use basically the pattern of debris around its nest to recognize where its nest is. It's just this little burrow in the sand that it has to find its way back to after navigating a mile away. So it can all do this with a very tiny brain comprising about 1 million neurons or so. The box jellyfish is perhaps even more amazing because it doesn't even have a brain. It has an astounding visual system. It has 24 eyes, and 2 of these eyes have excellent optical quality. And one of them actually has perfect optics, as is basically aberration-free. And so they've ascertained this. The one thing it might be using this visual system for is to navigate around within the river. So it basically hangs out around the river banks, and so it doesn't want to sort of be just in the middle of the river, and it's surveying the terrestrial landscape above it to understand sort of where to go to find its prey. Okay, so let's just talk a little bit about the jumping spider. This is just a depiction of the jumping spider visual system. It has a total of 8 eyes, which are shown here. 2 of those eyes, the ones in the center here, are forward-looking and have very high resolution, but basically tunnel vision over that very with high resolution. So basically over about a degree of angle, they have a very high resolution retina that scans back and forth, which I'll show in a second. And then these eyes on the side have kind of like a fisheye lens with very low spatial resolution, so it can survey things moving around it. And when it sees something moving or interesting that it might eat, for example, then it turns its head toward that. So these lenses are basically attached. They're fixed to the exoskeleton. It turns its head towards that so it can image it with its high-resolution eyes. And then the high-resolution retina is the back of those tubes, and it scans them back and forth to recognize the object. This is what the retina looks like. It's basically a one-dimensional strip of photoreceptors, about 5 photoreceptors wide and 20 tall. And it basically takes this strip and scans it back and forth at about 1 hertz to do object recognition. Okay, so it takes this spatiotemporal pattern of activity and somehow forms an image that it can use to do recognition. People have done jumping spider psychophysics and just shown that basically a jumping spider can distinguish the difference between prey and potential mate that it might exhibit courtship with. So that basically when a spider sees another spider as a potential mate, then it will do this little dance. They do these courtship dances. So these stimuli at the top elicit these courtship dances, and the ones that at the bottom elicit prey attack behavior. And those are the percentages they exhibit that with. And one of the testaments to their pattern recognition capabilities is that there's certain kind of flies out there that have developed patterns on their wings so as to fool a jumping spider. So it makes it look like another jumping spider that it might, mate with, I guess, instead of attacking it and eating it. So that's the fly on the bottom, and a real jumping spider is the top. Okay, so that's a kind of a testament. Nature's basically hill climbing in that space to find what is the optimal stimulus that can fool a jumping spider. And in fact, it's an image of a spider. Okay. So it's, and then this is basically just showing some of their navigational capabilities. A lot of these studies were done by Michael Land back in the 1960s, showing how they can orient to prey. If there's something moving out there in the environment, they're basically, they can sort of find the position of it with one of those low-resolution eyes on the side of their head and do this ballistic turn, and that's just showing the angle of one turn, not with like sort of incrementally going like, is it there yet, is it there yet, is it there yet? They just make one ballistic turn, and it's there. Okay, that's just showing the turn of that movement in response to the target in the environment. Okay, so, and then other people have studied their abilities to do navigation, so they can actually navigate in these complex 3-dimensional mazes. So what they do is they place the jumping spider on this pedestal here in the middle of one of these environments, and then the prey item is one of those 2 trays there. And so the jumping spider, first of all, it can't, those trays are too far away to jump at the item. And so what they notice what the jumping spider does is it doesn't just try to sort of jump initially and then fail or climb down the pole and just kind of randomly search around. The first thing that spider does on that pedestal is it kind of looks around its environment. It kind of just sort of looks around at things, and then it climbs down the pole, and it doesn't just sort of do a random search for one of the things. It actually finds its way to the correct pole and climbs up it to get the prey item. And to the right here is just shown the probability with which it does that. So for example, there was one at the rear, there was this one at the bottom, for example. When you put the prey item in tray A, it has to actually go back away from the thing it wants to get, right? It has to actually walk away from it and to the wrong side of space, right? So the prey item is the left side, it has to walk over to the right and then climb up that pole. And it actually does that. It doesn't just sort of go hunting around for the route. Okay, so there's some sense in which they can develop a sense of three-dimensional space around them to do navigation. These are some movies I made together with Wyeth Bear back at Caltech when we were grad students in Jim Bauer's lab using his dissecting microscope with a video camera attached. This is a one-day-old jumping spider we happened to catch because we found a pregnant female, and then just the day after it hatched, we were able to make these movies. So this is a jumping spider that's just have very little experience with the visual world, and this wire here this metal thing is a paper clip, just to give you an idea of the scale. And so these two black things here, the eyes moving inside the head, those tubes, they're scanning back and forth. And then this just exhibits some of their behavior, just kind of scanning around the environment. This is one of their jumps here. Okay. And then this is just kind of, this is very typical of their behavior. They do a lot of looking around and inspecting of the world, and they're kind of fun to play with. If you ever catch one of them, you can probably just catch them around here on the campus and bring them up to your computer monitor and weave around the mouse. They'll chase it, they'll try to attack it. They're very visual animals. They behave very similarly to a cat. Okay, and then here's a jumping spider tracking a Drosophila.
What's the size, like a millimeter?
This thing is, about a millimeter probably. It is an infant. We'll grow a little bit. Okay, so they do this tracking behavior. So you can I'll just do that again, right? So for all practical purposes, Christoph, I would call this visual attention. Maybe you can say it's conscious. Well, why not? So it's basically has some attention and ability to track these items in the environment, and it's doing all this with a brain of no more than 1 million neurons and a visual system probably of no more than 100,000 neurons. So basically think about a single hypercolumn of visual cortex, a V1, a single hypercolumn of V1 in this animal, and that's doing all of this stuff and after one day, after hatching out of the egg. Okay, so I think but the lesson I get out of all this after studying these tiny brains, going back to sort of the fundamentals of evolution, it sort of forces you to think about the problems that evolution had to solve when it designed brains, and what are these neural circuits really doing, and what do we build upon when we have these human brains. And this is a quote from Rodney Brooks, who's not someone I always quote, but I find myself agreeing with, but I definitely resonate with this statement here. I think it's very important, so I'll just read it out here. So he's saying problem-solving behavior, language, expert knowledge, and application and reason are all pretty simple once the essence of being and reacting are available. That essence is the ability to move around in a dynamic environment, sensing the surroundings to a degree sufficient to achieve the necessary maintenance of life and reproduction. This part of intelligence is where evolution has concentrated its time. It is much harder. Okay? So I think oftentimes when we do neuromorphic engineering, neuromorphic computing, AI, artificial intelligence, machine learning, and so forth, we're attacking these problems on the top. Right? We're trying to make things that play chess or do object recognition or speech recognition and things like that, or play Go. And we sort of think of these things, those are as the epitome of intelligence. And I think what Rodney Brooks is saying here is that these other things which we don't normally think about all the time as being really hard, just standing up without falling down or being able to open a door or something like that, these are maybe the more fundamental problems of intelligence that you have to kind of get right and build a system that can do that and do it robustly. And so this is kind of maybe to sort of think about as that some of the challenge problems out there that we should try to figure out how to solve with neural systems. And then maybe once we get those, move forward to these other ones. Or maybe they're not as hard, as like he's saying. Maybe they're not as hard as you think. Okay, so the second lesson I think I would like to focus on this problem of nonlinear processing in dendritic trees. So how do neurons really compute? And so I think what's kind of amazing is that we've learned a lot in neuroscience over the past 50 years about how neurons work, how they actually compute. But all this while, our model of neurons has basically stayed the same. So if we just kind of look back, in time, the neural networks that people were using in the 1960s, like Frank Rosenblatt and Bernie Woodrow, it was basically this model where we have a unit that linearly sums its inputs and then puts that sum through a threshold. They had developed learning algorithms for these neurons, shown they could train them to solve certain kinds of tasks, but they were stumped with a challenge of how to stack them into multi-layer neural networks. Even though Bernie Woodrow's learning rule was based on stochastic gradient descent, they were stumped by the fact that, well, if you stack these things, you have this hard threshold function. It's non-differentiable, so you can't backpropagate a derivative through that. So it took people 20 years to realize that, I guess, that you could make that function smooth and differentiable, and then you can do backprop and stack these things in multi-layer neural networks. So the thing that launched the second wave of neural networks in the 1980s, these neurons basically had sigmoid nonlinearities instead of hard thresholds typically. Okay, so that sort of survived for long times. And then more recently, sort of the deep learning revolution, it's basically the same model, but instead of a sigmoid, we sort of realized that, well, the problem with a sigmoid is that you're losing a lot of your dynamic range. You're just sort of clipping the signal at the top. And so there's really no reason to do that. And so you can have some preserve the dynamic range there, and it works better. Okay, so 50 years of evolution in our models of neural networks, and we've learned a lot in the meantime of how neurons actually work, and that's not that, right? So if there's one thing I think we know, is that neurons are probably not computing something like this, and I was astounded to hear Christoph this morning say that, well, the dendrites are, we're not going to use that so much in our models. And that's, to me, like Moses telling you that the 10 Commandments just don't really matter anymore, something like that. So I actually learned about all this stuff from Christoph when I was a graduate student. So anyways, there's certainly a lot to say about this. And so I think one model that people have talked about for these models going back a ways, so people like Jerry Feldman and Bartlett Mell back in the 80s and 90s, is this idea of a sigma pi neuron. So the idea that instead of just simply taking a weighted sum of inputs, it's actually taking a weighted sum of products of inputs. Okay, so it's taking local groups of inputs, multiplying them together, and then taking maybe a weighted sum of that, the so-called sigma pi model of the neuron. And that weighted sum of products is fed through a pointwise nonlinearity. Okay, but so the idea here then is where you impinge upon the dendrite makes a big difference in terms of impact you're going to have on that neuron. You can't treat the neuron as a single point node in a neural network, right? If you just make a graph of the connection structure from one neuron to the next, that's not going to necessarily tell you what you need to know. We need to know where is that input, what dendrite is it coming in on, and where on the dendrite is it pinching, and what other inputs is it combining with nonlinearly around it to really figure out what's going on. Okay, so Bartlett Mell has done modeling work suggesting that these neurons are perhaps better models as a multi-layer neural network, and one would have to sort of consider coincidences as well. And so that Paul Rhodes talked a lot about this morning earlier, the importance of incorporating these biophysical nonlinearities. And so Bartlett is really kind of led the wave on this, I think, in my view, and has just published really a lot of profound and very important papers over the years, starting with his work in Christoph's lab as a postdoc here on the sigma pi neuron, but basically trying to explore the computational properties, the computational richness of these nonlinearities in these neurons, and also basically comparing the physiology and showing that this can actually do a much better job modeling the physiology of actual neurons recorded in the brain. And so I just want to go back to this slide and just point out that, I think our inclination as always as scientists is to simplify as much as possible. And I think this is one place where we don't want to simplify, because oftentimes when you confront people with this, that these neurons are these nonlinearities, you say, well, I can make physiological measurements, and look, it actually kind of behaves linearly after all, and so we can all kind of rest easy with that. And that shouldn't really be our attitude here, because if anything, if this device actually behaves linearly, then it really begs more questions than it answers. It would sort of force you to ask, well, how is it that all those biophysical properties, those nonlinearities in the dendrites, how is it that they conspire together to make a linear system? You have another question to ask right now. And as well, from a computational point of view, nonlinearity is the essence of computation. So when you find nonlinearity in the system, you should be happy about it. You should be trying to figure out how it's being used by the system rather than trying to sort of push it aside. And I should also mention another person, Jeff Hawkins, has been doing some work incorporating these incorporating these dendritic nonlinearities in his models as well. So I think there's just tons of work to do there. It's totally unexplored, and I think biology is giving us a very strong lesson, a very strong cue there, hint, about how to build computational systems based on neurons. And it's not simply a weighted sum of inputs, but exploiting these nonlinear properties. And the third and final topic is this on feedback. And to sort of just introduce that, so the basic idea here is that feedback loops are pervasive all throughout the brain. We find them everywhere. And so what are they being used for? And so this is maybe just one motivating example of that. So how many people, don't say if you know what this is, don't say, but just raise your hand if you can see what's in this scene. It's an animal. Anybody see it? So one? Oh, somebody said it. Shh. Okay. Two people see it. 3 people see it. Okay. So there's an image of an animal in this scene. It's kind of like that image of the Dalmatian dog, but the difference is you can use this because not many people have seen it before. So if you ask people, who not seen this before, who don't know what's in the scene, if you ask them to draw what they see in the scene, this is what they draw. Okay, so this is what I would draw, I suppose. If you just basically you draw the edge map of the scene, right? You just draw the edges in the scene. It looks like nonsense. Okay, so I'm going to tell you then what's in this scene. It's an image of a cow, and it's facing towards us, and this part at the bottom, that's the nose, that's the snout of the cow, and the eyes are sort of in the middle there. Does anybody see it now? So raise your hand if you see a cow. Some more people see a cow. Okay, wow, okay, great. So now you take the person who sees a cow, and you ask them to draw what they see, and this is what they draw, okay? And so it's like, what? This is the same image, right? And they're putting lines in here where there are no lines in the image. I mean, there's, where most of that face there they draw in, there is no evidence for that at all, right? There's no edge map. There's no edge there in the image. The edge map is on the right, okay? So where'd this come from, right? This came from inside your head. So it's basically a hypothesis that you have about this scene that you're seeing. And moreover, you're not just sort of outputting some category and saying, oh, cow, right? I mean, you actually can kind of see what kind of cow it is, the texture. You can kind of explain away all these sort of detailed graininess of the image and so forth. And so how's that happening? And so what, we have this fact that the way the system is constructed, it has all these very strong feedback loops in the system. And this is showing the architecture of the visual system. As you go from lower levels to higher levels, there's a feed-forward pathway, but also a very strong feedback pathway. There's strong feedback connections, recurrent connections within areas, and also between the thalamus and the cortex. There's this very strong feedback loop. It's deeply mysterious what the pulvinar is doing. It has multiple maps of visual space. It co-evolved with the cortex. That's a massive structure within the thalamus that interconnects with the cortex. We haven't a clue of what it does. Oh, actually, we have a clue, but very, very limited understanding. And as well, within any given visual area, there are these strong recurrent loops within a visual area. So the neurons within V1 are connected in feedback loops within V2 and so forth. So what's all this doing? So in terms of a theory, so I think one promising theory that's been put forward is by Tyson Lee and David Mumford, this idea of hierarchical Bayesian inference. So their idea is basically that you have this kind of hierarchical system that's trying to come up with a hypothesis of what's going on in the scene. And higher levels are basically sending their predictions to lower levels about what they think is going on. They're saying that they're sending a message which is, okay, I think I'm seeing a cow. If I were seeing a cow, this is where I would expect the edges to be, right? Please see if you can find me an edge perhaps in that part of the scene. So these higher levels like V3 and V2 are making predictions down to V1 to reformulate the information there, and then as a result of that, the representation in V1 is actually going to change. Okay, it's not going to stay static. It's not just a slave to the image. It's going to be modified as a function of what higher levels are saying to be more consistent with these hypotheses. Okay, so I think that's one very promising theoretical idea that's out there about what these feedback loops may be doing. So the biology is just telling you in spades that it loves feedback loops. It just puts feedback loops everywhere. It's corrupting the data with feedback. Even at the very beginning in the retina, the photoreceptors are subject to feedback from the horizontal cells. So the photoreceptor is not giving you a vertical measurement of how much light is coming in. It's already the output terminal, the synaptic output of a photoreceptor is modified by context of what's in the surround via the horizontal cells. Okay, so that's basically the theme throughout biology. And I think we as engineers and computer scientists, we need to figure out how to harness how to harness these feedback loops in the system rather than being afraid of them and not using them. Okay, so I think what we should be leading towards, with the sort of standard kind of idea now is on the left, just sort of this feed-forward propagating stream. We should really be thinking about, well, how would I use these sort of bidirectional flow of information to come up with an inference of what's in a scene. And the difference here is, rather than just kind of trying to discriminate things that we're trying to sort of say, is it a cat, is it a school bus, or whatever, we're trying to actually sort of come up with some explanation of the underlying causes, a model of what's in the world out there, a model of the environment. And this, we're constantly sort of, we have a hypothesis that we're trying to test against the data, as opposed to doing classification. Okay, so I'll just leave you with that, and if you're interested further in these ideas, I've written a number of papers on them, and I'm happy to take questions. Right. Well, thanks as a very important question, because I mean, I think one of the arguments here is that we can't just do biological mimicry, right? We need to be informed by the theory at the same time, and I guess my sort of philosophy is turned toward instead of saying, well, it's irrelevant until proven relevant, I would say everything is relevant until proven to be irrelevant, okay? So it's potentially on the table, and that's going to drive you crazy, right? Because there's so much detail, then how do you sort through it, right? But I guess I would zoom in on things like dendritic nonlinearities, because you know they're there, they're potentially useful, right? We have hypotheses that can sort of, like things that Bartlett-Mell has done, showing how they potentially can be exploited. And so I think that's maybe your trail to chase. Yeah. Okay. Yeah. Well, okay. So the test is, I guess, can you harness that to do something useful and interesting, right? And then I think that puts it maybe sort of EPSIS level on the table of hypotheses space. But at first you're sort of like, well, gosh, everything is potentially a game here. But I think to say, look, if you can show the stochastic resonance property can be used for something, if it can actually solve a problem and do it efficiently, then I think that's interesting to start thinking about. But yeah.
So there's a way. So there's a way to actually go about that. I think that would satisfy the parsimony that you want to get to, but still respect the neural mechanisms that we're talking about, and that is to put and you have to start somewhere with a circuitry of some sort. It has to do something. It has to perform some sensory processing or sensory motor function where the performance can be quantified. Then you have a palette of mechanisms, not 1,000, but let's consider, 4, 6, 8, 10 of these neural mechanisms that we're talking about, each one of which has its own parameters. And then one can evolve such a system with a complexity constraint. And then you see whether mechanisms like synaptic depression, in the case of object recognition and different sensory modalities, whether these mechanisms survive in an artificial evolution process. So, so there is. That's one example of a way to tackle exactly the question that you're raising, and incorporate the mechanisms that we all feel are ubiquitous and therefore probably computationally relevant.
Okay, so.
Any more questions? Actually, Jeff.
I think this is a great talk, Bruno. I really enjoyed that. I think this question that Christoph brings up is really, really central to so much of what this room is about. And I know I've had conversations with numerous people in this room, modelers, people building hardware, about the importance of active dendrites. I think it's worth a little bit more conversation here. I just want to put one more point on the argument in favor of them. I think when we think about neurons and we think about pyramidal cells throughout the cortex or anywhere in the brain, if a cell has 10,000 synapses, and we know those synapses are expensive, they are connections, they represent dendrites and axons and energy, they're really expensive. We have to ask ourselves, what can I do with 10,000 synapses on a neuron? If you do not accept nonlinear properties in the dendritic trees or nonlinear properties in the neuron itself, it's very hard to come up with an explanation that justifies the 9,999 synapse. If you can group those synapses into small groups, 20 to 40 at a time, that act as independent pattern recognizers, as Bartlett, Mell, and other people have argued, then all of a sudden you've got a neuron that makes a hell of a lot of sense. And Christoph, I think the reason we have to pick things to care about is we have not just we can do something better with it, it's because we have a huge mystery to explain what the hell a neuron is doing with 10,000 synapses. And if we find a really logical explanation for it, we should pay a lot of attention to it until we find a better one. And that's my argument for that particular nonlinearity that Bruno was arguing for, and I like as well. Thanks.
Thanks.